Good evening, everyone. Thank you all for attending our seminar today. I have the pleasure of introducing Dr. Michael Golding as a speaker for this seminar presented by the Michigan Center on Life Stage Environmental Exposures and Disease. Dr. Golding is currently a tenured associate professor in the Department of Veterinary Physiology at Texas A&M University and trains his laboratory students to challenge specific societal norms in their developmental programming research. His most recent research and the topic for this seminar today focuses on questioning traditional paradigms on pregnancy and development. His team is trying to understand how male drinking prior to conception contributes to development of alcohol-induced birth defects and later life disease outcomes. Please join me in welcoming Dr. Golding.
All right, thank you. So first, I'd like to thank Pini and Vasantha for the invitation to speak here today, as well as for the rest of the program and taking an interest in the work that we're doing, trying to understand how male environmental exposures are potentially influencing fetal development, and then how this might explain the birth defects associated with fetal alcohol spectrum disorders. Okay, so to begin today, I'd just like to point out this rather interesting paradigm here, that when a couple is getting ready to have children, we send the woman in for her prenatal medical visits. We provide her with extensive counseling on the dangers of smoking, drug and alcohol use, and we even make sure that she's eating her vegetables and taking her multivitamin. In contrast, the lifestyle choices of the father and paternal preconception health are dismissed as irrelevant. And so I would argue that the misconception that sperm only transmit genetic information and nothing else has resulted in a major gap or a blind spot in our understanding of both developmental toxicology and the development of pediatric disease. And so we've known for several years now that sperm contain epigenetic information that is vital to the establishment and maintenance of a healthy pregnancy. And so on point, this is actually how imprinted genes were first discovered. However, despite this broad recognition that the sperm inherited epigenetic program has a major role in directing development and differentiation of the embryo, birth defects, in particular congenital defects associated with fetal alcohol spectrum disorders, are still exclusively blamed on maternal exposures. And so one of the very first grants that I ever received was from the National Institute of Alcoholism and Alcohol Abuse. And like all my peers, I examined maternal models of exposure. However, once I started getting into the literature, it quickly became clear that we were probably missing half the story here. So in point of fact, about 75% of FASD children have alcoholic fathers. And if you look at the literature examining male alcohol use disorders, male alcohol consumption vastly exceeds that of females. And so what I aim to do in this talk here today is to challenge the prevailing paradigm that fetal alcohol spectrum disorders are exclusively the woman's fault, and demonstrate that at least some of the symptoms are attributable to alcohol use by the father. And then at the end, I'm going to go into some of the mechanisms by which that memory of exposure transmits across to the offspring. Okay, so to begin today, I'd like to talk broadly about how epigenetic mechanisms contribute to toxicity and the development of birth defects. And so at its essence, epigenetic mechanisms are the biochemical means by which cells retain a memory of past experiences. And so that is to say that they retain information from past experiences for the purpose of influencing future actions, be it gene expression or developmental programs or morphogenesis or what have you. And so the term epigenetics is now widely used to describe just about anything in biology. But to me, I like this definition here the best. And so epigenetics are heritable, non-genomic mechanisms that enable cells to retain a memory of past states and perturbations. And so this work is done or exemplified by some studies that were done, say, by my former advisor, Mark Westhuizen, looking at the process of exon activation in clones. And so here we have the donor clone here, this calico cat, and then here's the cloned over here, which is just this gray one. And of course, these cats had undergone random exon activation, and so that's why we have these different coat color patterns. And also work done by Barbara McClintock looking at the color of corn kernels. And so both of these examples here, provide evidence of epigenetic changes driving visually identifiable phenotypes. And so be it an epigenetic change that resulted in a random process of exon activation, or something that could be tied to stress, as in the case of the changes in the corn kernels. And so during differentiation, the nucleus of each cell becomes organized into domains of co-expressed or co-repressed genes, and each of these active or silent domains then is characterized by unique patterns of DNA methylation or various changes in histone structure. And so the organization of these domains becomes tissue-specific during development. These changes facilitate transcription, but importantly, these processes are extremely susceptible to environmental influences, where changes in nuclear structure can then lead to differences in gene expression that then persist across the life stage. And so recently we've started to look at epigenetic changes in nuclear organization to help explain how early life stressors or exposures to various things, be it nutritional deficiencies, exposures to drugs of abuse or environmental chemicals, how these things then might influence epigenetic processes and then go on and cause developmental defects either shortly thereafter or much later in life. And so what we're doing here broadly is changing our definition of toxicity to move away from this idea where we've been focused for perhaps the last 100 years on acute changes, where you have this idea that the toxin comes in, it does something, poisons the developing embryo or conceptus, and then you have an immediate response. And so what we're starting to shift towards is this understanding here that these stressors or these chemicals can then come in, modify chromatin structure. This then imparts a memory which persists across the life stage and then might result in disease, development of disease much, much later in life. And so building off this perspective here, my group decided to ask the question, can paternal drinking influence fetal alcohol spectrum disorder phenotypes? Is there an epigenetic means here by which the memory of a paternal exposure to alcohol persists through sperm and then starts to influence the development of the conceptus? And so before I start going into our data, I'd like to just introduce the team that actually did the work. I'm essentially just a glorified spokesperson here. And so the work I'm going to be talking about today, much of the published work here was done by Richard Chang. A lot of the unpublished work that we're going to be looking at, placental changes, is going to come from Kara Thomas and Yudi. And my technician here, Katie, is one of the people who was extremely instrumental in getting the micro CT analysis of placentation up and running, which I'm going to talk about here in just a second. Okay, so the core defects of fetal alcohol spectrum disorders are these. You have placental defects and fetal growth restriction. This growth restriction then persists and into the postnatal stages. And so these kids are basically growth restricted on into postnatal life. And then you also have some craniofacial defects. And so these are the visible phenotypes that we can see. And of course, there are going to be some accompanying central nervous system dysfunction that goes along with these changes as well. And so the question that we started with was, can paternal drinking influence the emergence of some or perhaps all of these classic, core FASD phenotypes? And so then the model that we used was a mouse model of what we call the limited access model, which is a model where you have the mice, their water bag is basically exchanged for a bag containing 10% alcohol, and then the mice have limited access to this, this alcohol for a very short window. So basically, as soon as they go into their active cycle, we pull the water bag off, put the alcohol in there, and we leave it in there for 4 hours. And so this model is really meant to mimic a short-term exposure, a daily short-term exposure, such as you would have, say, of someone who goes to the bar immediately after they get off work, or some other kind of social circumstance like this. And so what happens here is that this is a voluntary model, and so the mice are not forced to drink, and so the blood alcohol levels are relatively normal. And so that means that there's somewhere, just say here, around 120 milligrams per deciliter, which, just to roughly translate this, is about 1.5 times the legal limit. So certainly you're not blitz, but this is nonetheless, you've had evidence that you've been drinking. And so as soon as we get the mice and get them on the alcohol and they start to consistently drink, we keep them on this exposure paradigm for 70 days. And so this in mice represents 2 complete spermatogenic cycles. And so we did this to ensure that the premeiotic and postmeiotic sperm that were produced are all exposed to alcohol. And that way we can, eliminate any kind of confounders of sperm that were made before the alcohol exposure. And so we did this because we don't know when the epigenetic memory of alcohol exposure might be imparted. So we basically just encapsulated the entire spermatogenic cycle. And so after we've had this exposure for 70 days, we then would mate the males at least 3 times in order to get a good number of pregnancies. And so this model has been extensively described in these previous publications. And so I'm just going to go through moderately quickly here some of the data that we've seen so far. And so the work I'm going to be talking about here is looking at gestational 16.5, which is this phase in development where you really start to see a dramatic increase in fetal weight during the last phases of pregnancy. All right, so just before I get into that, I want to emphasize the clinical relevance of our model. And so I pulled this figure from a book by Tim Cook, which examines the economic impact of alcohol exposure, actually. And so what he found here was that the top 20% of alcohol consumers would, at least get 2 drinks per day. And so this is the top 20% right here. And so in this one group right here, you can see that they're basically having about 14 drinks per week or 2 drinks per day. Whereas this group right here, these are the hardcore drinkers right here. And so this top 20% here corresponds to about 10% of all males between the ages of 18 and 50, excuse me. And so this model is one that is widely applicable, I think, to our society, because there are indeed a large number of males who drink. And as I mentioned at the beginning of the talk, male alcohol use dramatically outpaces that of females. Okay, so to begin with here, the first thing that we did was to start looking at the influence of alcohol on male reproductive physiology. And so what we saw here, this is just a cartoon diagram right here looking at the male reproductive tract. And when we went in here and looked at the weights of the testis or the epididymis or any of the other accessory structures here, including the seminal vesicles, we did not see any alcohol-induced changes in the weight or the morphology of these structures. And then similarly, when we went into the testis and started doing histology, we did not see any differences in the seminal vesicles in terms of their surface area, a number of cells, or any other parameter that we could measure. And there was no overt change in circulating levels of testosterone. So then we started looking at the sperm themselves, and we saw that sperm counts were the same between the 2 treatment groups. We looked at the percent tunnel, or how many sperm were undergoing apoptosis, and there was no changes there. And then similarly here, we also used this CMA3 stain, which is one that's going to intercalate in between DNA. And so this is really a measure of chromatin compaction. And we did not see any alcohol-induced changes in chromatin compaction. And then similarly here, you probably would, as you expect, there was no difference in litter size in the next generation, and there was no differences in the distribution of males and females in these respective litters. Okay, so moving on to the offspring. And so here again, we're looking at gestational day 16 and a half. And so we have three measures here. The first one is the weight of the gestational sac. And so this is a complex of the placenta, the amniotic sac, and of course the fetus inside. And we saw here that both male and female offspring exhibited decreases in the gestational sac weight. We then looked at fetal weights, and we could see here that again, in both males and females, we saw a decline in fetal weight, and there was an equivalent or corresponding, I should say, decline in crown rump length. And so the thing that was really kind of interesting to us was the placenta. And so what happened in males was that we saw an increase in placental weight. So this was certainly different than what I had expected, because generally speaking, in other mouse models of gene knockouts or what have you, you generally see parity between fetal weights and placental weights. So a small placenta would equal a small fetus. But here we are seeing an increase in growth in the placentas, and then here in the females, there was actually no difference. And so we've been studying placentation in mice for a large number of years, and so we now have a pretty good mathematically quantifiable understanding of the rates of growth during mouse development. And so the graph I'm showing you right here is one I stole from a paper from Mu et al. In 2008, and you can see here that placental growth really starts around gestational day 10, and it's going to peak right here around gestational day 16, 16 and a half. Whereas you can see fetal weight kind of goes through this lag period here, but then around 14 and a half starts to turn on the jets and goes forward towards birth. And so you can, at each developmental stage here, take a ratio between placental weight and fetal weight and calculate what's called placental efficiency. And placental efficiency at its essence is how many grams of fetus did you get per gram of placenta? What's your bang for your buck here? And what we saw here with that increase in placental size and the decrease in fetal weight was that you had this rather precipitous loss or decline in placental efficiency. And so this broadly told us that the placenta, even though it had formed, was not working at optimal levels and was perhaps not working as it should. And so then we move forward to start looking at these animals and following them into postnatal life. And what we saw first was that the growth restriction that we identified during gestation persisted at birth. And so we can see this data here. These animals are about a week old. And then when we started to follow these offspring, this is also kind of interesting in that the females were rapidly able to catch up to the control group, whereas the males continuously lagged behind. And so we had this sex-specific observation that started to emerge where the males were really horribly impacted by this preconception exposure of their fathers, whereas the females were able to shake it off relatively easily, at least according to this one measure. And then when they reached adulthood, we started to look at just basic measures of their metabolism. And what we saw was that the males had lower blood sugar levels. They were insulin hypersensitive, whereas females started to exhibit signs of insulin resistance. And so these findings have been published, and they're described in Richard's paper, Chang et al. In molecular metabolism. And so if you're a metabolism-focused person, I encourage you to go read that paper and where we start to go into the mechanisms that underlie this. Okay, so moving back to our core goal here of discussing the influence of male drinking on the emergence of FASD defects, we can see here that yes, we can see placental defects, and these associate with fetal growth restriction, and that at least in the male offspring, we also see postnatal growth restriction. And so the last phenotype that I want to focus in on here is the emergence of craniofacial defects. And so for this study, this was work done by my student, Kara Thomas. And so she decided that she needed to have a control, an appropriate control here. And so she made up this 4x4 factorial design here, where we looked at the offspring of parents who are just exposed to water or to the control treatment. What would happen if, mom drank? What would happen if dad drank? And then what would happen if you had this dual model of exposure? And so again, we exposed the males to alcohol for this 70-day preconception period. But for the females, we began exposing them to alcohol for 10 days prior to exposure, and we continued the alcohol exposure until gestational day 10. And so this is right around the point, just a little bit past gastrulation. And so this is kind of equivalent to what we see in clinical cases where a woman who having an unplanned pregnancy would realize that she is pregnant and then stop drinking. And so we pulled them off alcohol at this point, pulled the dams off alcohol at this point. And then again, we went out to gestational day 16 and a half. And so I want to make an important point here, especially with relation to, how we did this work. And this is going to lead into a discussion of cage enrichment. And so when we first started these experiments and we pulled the females off alcohol, they went through withdrawal. And so they had, increased stressors. We had a large number of pregnancies that we could establish. And then, of course, when we pulled them off alcohol, we lost those pregnancies. And then we also have some postnatal studies that I'm not going to be discussing here today. But as soon as those offspring were born, the dams would immediately start to cannibalize them. And so we saw this seminar from this a veterinarian, I think from UC Davis, who described how important cage enrichment was. And so what we did was we went in and added these little igloo huts, which you can buy from a company that specifically makes cage enrichment. We had an overabundance of nesting material, and we also got them these gummy bones and these little kind of wood gnawing sticks, something that they could occupy their attention, making their nests really robust, as well as just gnawing on these sticks to kind of relieve anxiety. And when we did these changes, these relatively simple and small changes, we had a dramatic increase in pregnancy rates, a dramatic increase in the number of live births, and the problem we had with cannibalism virtually disappeared. And so a very important point here, I think, that you need to consider if you're going to be developing experimental models that stress out the dams. Okay, so moving forward here to look at craniofacial development. Fetal alcohol spectrum disorders are characterized by a smaller head. You have changes in the spacing between the eyes, as well as the overall size of the eyes. And so we went into the mouse and we started measuring equivalent measures. And what we saw was that the size of the eyes decreased. And so we could see this in males. It appeared in the paternal model of exposure and the dual model of exposure, whereas in females, we could see it in the maternal model of exposure as well as in the dual model of exposure. So that being where both parents drank. Then we started to look at the distance between the eyes. And so this is really where our model really started to find some interesting data. And so what we saw here is that regardless of whether mom drank, dad drank, or it was both parents who drank, we saw an increase in the size, the distance between these eyes. And then finally, we looked at what we call mid-facial depth. And so this is just a measure of how flat the face has become, which is kind of equivalent here to the structural development of the human nose. And so what we saw here was that the male offspring of alcohol-exposed moms, alcohol-exposed fathers, and then the dual parental model, these also all displayed changes in mid-face depth. All right. And so the question that we had asked at the beginning of these experiments was, can paternal drinking influence fetal alcohol spectrum disorder phenotypes? And I hope I've provided some convincing evidence that yes, we can. So we do indeed see placental defects and fetal growth restriction, postnatal growth restriction, and we're now starting to see some strong evidence of craniofacial defects emerge as well. All right, so then the next question that we wanted to ask was, does the maternal background influence the penetrance of paternally inherited epimutations? And so this question is one that we'd thought about for a while. And the basis of it is this, that in mouse models of FASD, there are certain strains that are resistant to developing the teratogenic effects of alcohol. So for example, if you expose C57 Black 6 J strain mice to alcohol, their offspring develop defects, whereas in the CD1 or another outbred strain of mice, these mouse models do not. And so we wanted to ask the question, rather than say from the point of view of an acute toxicity, is there a potential here that maternal background could alter the penetrance of these paternally inherited epigenetic changes? And so again, this is work done by Kara Thomas. And so what we did here was to take the alcohol exposed males. They again had undergone this exposure paradigm here for 70 days prior to mating. And then we mated these or bred these males to CD1 dams. So we chose this strain here because it's an outbred strain, and they were available in our mouse facility. And so this was kind of just an interesting, a quick experiment, or we thought it would be, just to see, what would happen. And so what I'm going to do here is to start to show you the data from the pure C57 Black 6J. So this is the strain that I've been using all along, and to the data that I've presented to this point. And then over here on the right-hand side, I'm going to start to present to you the hybrids. And so you can see here that we saw growth restriction in both the male and female offspring of these, the pure C57 Black 6. But when we examined the hybrid offspring, what we saw was that growth restriction went away. And then when we moved on to placental phenotypes, we saw again here, when we repeated these experiments, that we had this, again, the placental overgrowth in the male offspring, whereas the female offspring were identical to the controls. And then if we looked over here in the hybrids, you can see here that the maternal the breeding of the alcohol-exposed males to the CD1 dams, this resulted in a decrease in placental weights. And so in the pure C57 Black 6, we have an increase in placental weights in the male offspring. Over here in the hybrids, we have a decrease in placental weights in the offspring. And so again, when we come back to that placental efficiency, we can see here that again in the C57 Black 6, we lose placental efficiency, or we have that decline in placental efficiency. Whereas over here in the hybrids, we had a trend towards increased placental efficiency. And so the p-value here was 0.08, which for my money is close enough to p05. All right, so this suggested that indeed maternal background can modify the phenotypes that are programmed into sperm. And so then the question became, well, what was going on here? What was the actual histological changes that were behind these, kind of overt macro measures? And so to ask this question, my technician, who had a large amount of experience using micro CT to study bones, she actually found these protocols online where you could stain fetal tissues, particularly the placenta, with phosphotungstic acid, and then stick these tissues into a micro CT imager. And then once you had those images, you can go in and start to dissect out the different layers. And so rather than doing histology the old-fashioned way, making sections, what this allowed us to do was to go in and to take 3D measures of these tissues, and then go in and find the volumes of the different layers. And so we're going to look here at the junctional zone, which is on the outside of the placenta. You've got the labyrinthine layer right here in the center, and then you've got the chorion, which is this portion I've marked down here in red. And so again, here are the different structures represented, in a cartoon diagram. And when we started to look at the changes in volumes in the different layers between the treatment groups, what we saw is that both the male and the female offspring had decreased size of their chorion. So that's this layer right down here. And then when we started to look at the junctional zone, we saw that it was the females that exhibited a decrease in the junctional zone. So again, the females did not display any overt changes in placental weight, placental diameter, or even placental efficiency, whereas the males here exhibited no histological changes whatsoever. And then similarly here, when we started to look at the labyrinth layer, what we saw is that the males did not exhibit any changes whatsoever, whereas the females had an increase in the size of the labyrinth layer right here. And so this is a layer where you have this exchange occur with a large number of blood vessels. And so when we started to look at these tissues, we could see that there was a lot of what my technician called negative space. And so this suggested that there was some increased cell volume in the decidua. And so when she went in and did the old-fashioned histology here and stained with periodic acid Schiff, and this stain here then goes after and marks out the glycogen-containing cells. What we saw here was that the placenta is derived from the female offspring of alcohol-exposed fathers, had this really large increase in glycogen cells, whereas the males did not. And so these data here suggest that there's some pretty significant sex-specific changes going on in terms of not only how the phenotypes manifest between males and females, but in terms of the histological organization of the placenta. And so the question here of, does maternal background influence the penetrance of these paternally inherited epimutations? I think the answer is yes. It does modify these placental phenotypes that are induced by altered epigenetic programming in the male. And then the other interesting thing to take away from this is that the female offspring seem to exhibit evidence of placental adaptation, whereas the males appear to be much more flat-footed. They don't really seem to have the ability to change dynamically their placental layers or to increase the glycogen reserves that they would need, at least on gestational day 16 and a half. And so these studies are just starting to be wrapped up now and sent out the door for publishing. And so then the question that we wanted to chase these observations with was, well, what molecular changes are underlying these phenotypic alterations? And so this work, what we did was we went back to the pure C57 black 6. And so this data I'm showing you right here is actually looking at gestational day 14 and a half. And so we went back here because this is the developmental stage where we can first detect changes in placental efficiency. And so the males don't quite exhibit it just yet, but the females do. And so what we did here was to get these samples and to then do RNA sequencing and to start to look to see what differentially expressed genes we could identify. And so we found a decent number, I think 300 to 500 differentially expressed genes. But the interesting thing here, at least to me, was not the fact that, or the individual genes themselves per se, but it was the expression pattern between males and females. And so what we found here was that, broadly speaking, males had a loss of expression, whereas the female offspring, for the exact same cohort of genes, upregulated these particular genes. And so we had this diametrically opposite change in gene expression between males and females, but for the exact same cohort of genes. And then the other thing to emerge from these studies was that a lot of these genes had the same address. And so when we started looking at their location, their chromosomal location, they all fell within, or not all, but a large number of them fell within gene clusters. And so the data I'm showing you here are the genes that we identified within the albumin cluster, the apolipoprotein clusters, and then in the fibrinogen clusters. And so what you can see here in the male offspring, which exhibited downregulation of these transcripts, was that if you looked across the gene clusters, we saw broad suppression of gene transcription. And so the only outlier here in this cluster was the ApoA5 gene, And when we started to dig into this and try and understand why this one gene here would be left alone, we discovered that this gene here was not regulated by the enhancer element that is central to the regulation of gene expression in this cluster. And so this suggested to us that there might be something going on here with chromatin architecture. And so we've known for years now that chromosomes are not arranged linearly within the nucleus, that there's a large number of chromatin architectural factors that then help to organize the genome into these three-dimensional loops. And so one of these factors is CTCF. And so what CTCF does is it essentially grabs different regions of the chromosome and then pinches them together in order to make a chromatin loop, right? And so what happens here is that these two structures can then be joined by bridging proteins that then allow that chromatin structure to form these organized loops. And in doing so, it basically serves as a boundary function here, keeping repressed chromatin on this one side and allowing for this loop structure to be opened up into a chromatin conformation that would then facilitate transcription. All right, so then the hypothesis that we had here was that that CTCF and chromatin looping might be disrupted. And so what we did then was to go forward and start doing chromatin immunoprecipitation followed by deep sequencing on these placental samples. And so when we did this, we had very, very good clustering here in our principal component analysis. So that means that the control samples here moved very far away from our alcohol-exposed samples. And when we look to see at the distribution of CTCF across these samples here, we found that we had a large number of CTCF binding sites that were shared between the control and alcohol groups, but we also had a large number that appeared only in the alcohol-exposed group and some that were lost in the alcohol group and only appeared in the control preconception treatment group. And then when we started to look to see, was there a relationship here between CTCF binding and differential patterns of gene expression, we found that about 25 to 30% of the differentially expressed genes that we had identified had altered enrichment of CTCF either within their gene body or towards their promoter regions. And then when we went back to the gene clusters that we had identified earlier, specifically the apolipoprotein, the albumin, and the fibrinogen gene clusters, we again saw that the decrease in transcription or gene expression coming from these clusters was accompanied by drops in the enrichment of CTCF across these loci. And so this suggested to us that perhaps the memory of alcohol exposure was persisting through sperm and in some way, shape, or form modifying gene expression patterns in the offspring, such that you had these really kind of interesting sex-specific changes occurring to a common or a shared cohort of genes, and that these changes in gene expression seem to fall within gene clusters. And this again correlated with changes in CTCF, or the chromatin architectural protein, the one that's going to be facilitating chromatin looping, CTCF, changes in the location or localization of that particular protein. All right, and so then the last question I'm just going to ask here today is, how is it that the memory of paternal alcohol exposure transmits to the offspring? Okay, and so broadly speaking, sperm are able to transmit epigenetic information through patterns of DNA methylation, sperm-inherited non-coding RNAs, as well as the region-specific retention of histones. And so we began by looking at changes in DNA methylation because there was a large body of literature out there that suggested that there was an interaction between alcohol exposure and the one-carbon metabolism pathway, specifically the generation of S-adenylmethionine. However, when we looked at this, we did not see any differences in the males in terms of their levels of overall SAM. And then similarly here, when we went and looked at the methylation profile of sperm, there was no global changes in DNA methylation either for the CpG or other contexts. And when we did the bisulfite mutagenesis and deep sequencing, we had virtually no changes come out. I mean, there were a few, I think something like 25 loci that had very modest, let's say 5 to 7% changes, but they were in cohorts of genes that really did not make any sense. Some of them involved in, developmental processes that are way further along than, into adulthood. And so this really didn't make sense to explain why we were seeing changes in placentation. Okay, so then the next element I want to discuss very briefly here are sperm-inherited non-coding RNAs. And so there's a large body of work produced, some of it by Oliver Rando's group, that has demonstrated that as sperm travel through the reproductive tract, the epididymis, that they exchange or pick up different cohorts of non-coding RNAs. And so, you start off with a highly enriched profile here with Pee-wee RNAs, but gradually these RNAs get shed, and the enrichment of tRNA fragments is increased as sperm move towards maturation. And so we sequenced the non-coding RNA profile of alcohol-exposed sperm. And what we found was that we could identify some shifts in both piRNAs and tRNA fragments. And if you took a ratio of these two, similar to what had been suggested in that previous publication, that the tRNA fragments represent the more mature non-coding RNA, and the piRNA fragments represent the more immature non-coding RNA. Well, what we saw was that the sperm of alcohol-exposed males seemed to have a decrease in the non-coding RNAs that were associated with mature sperm. And then when we sequenced these samples and identified individual microRNAs that were differentially enriched, and then compared this with the microRNA profile taken from maturing sperm, again, we found that a lot of the non-coding RNAs that were differentially enriched were ones that change dynamics during epididymal maturation. So this suggested that there's some changes occurring here that are primarily happening during epididymal maturation. And so I apologize for going through this relatively quickly. These data are published, and so if you want to go get them, UD's papers in reproductive toxicology. All right, so the last thing I'm going to speak about here is the region-specific retention of histones. And so as sperm are produced, the large majority of histones are evicted, and they're replaced with these proteins right here, these protamines as they're called. However, there's certain regions of the genome that retain histones. And so these regions are generally thought to be on the promoters of developmentally crucial genes that are going to be necessary to initiate the embryonic program. And so when we did some Western blot analysis, just looking between the differences between the enrichment of histone, and this one here is histone 3 lysine 4 trimethylation between alcohol and control sperm, what we saw was that there was an increase in the amount of this particular chromatin modification in the alcohol-exposed sperm. So again, we did ChIP-seq analysis. And so the interesting thing here was that these samples all piled up on top of each other. And so I apologize for this rather poor graph. These data are kind of fresh off the press, and so I haven't had a chance to make the symbols align with each other. But essentially what we found, or what explains this lack of clustering, is that the alcohol-exposed sperm contain a large number of additional peaks in over top of the ones that we would normally see here in the controls. And so it basically you just have 2 or 3 times the amount of H3K4 in the alcohol-exposed sperm. And so when we did the comparison between these samples to see where it was that these differentially enriched histones were binding using a chip seeker, what we found was that there was a rather large increase in, decrease in the amount of distal intergenic enhancers that were identified in the alcohol-exposed sperm. So even though the peaks that were gained in the alcohol-exposed sperm were more abundant, there seemed to be fewer fewer distal enhancers identified in these samples. And so you can see here the proportion of blue, which represents promoter regions, and the proportion of brown. If you look at those samples that are found only in the control or that are lost in the ethanol, that there seems to be more distal enhancers that are missing in the profile of the alcohol-exposed sperm. And so when we take these chromosomal regions and we use them to identify different genes or particular pathways, what we found was that a lot of these genes seem to converge on developmental processes, including organism survival, nervous system development, and embryonic development. And so just to kind of bring this all home, what we saw here, and I'm using some of the genes that I pulled from this data set. Here we have DNA methyltransferase 3A and some of these lysine demethylases. We could see that the profile of H3K4, excuse me, this is wrong, H3K4 in the alcohol-exposed sperm was higher for these two genes here, but then lower. And then these three genes also came out of our placental data set looking at CTCF enrichment. So there's some suggestion here that alterations in H3K4 trimethylation might be linked to some of the changes that we saw in the placenta with CTCF. All right, so just to kind of summarize things here, changes in sperm non-coding RNAs and histone structure correlate with offspring phenotypes. Alterations in the localization of chromatin looping protein CTCF associates with altered gene expression across gene clusters and also gives us this very interesting sex-specific change. And then these differences in gene expression then are correlated with the loss of placental efficiency that we saw, as well as some of the histological changes that we saw in the offspring of alcohol-exposed males. All right, so that basically concludes the presentation that I had for today. And so I'll just like to again acknowledge the students that did the work. So a large number of the placental work was done by Kara Thomas, and then the sperm-focused work and also the placental CTCF ChIP-seq was done by Yudi. And so then the last slide I have to present here, I should acknowledge my funding. I am supported by grants from the National Institute of Health, National Institute of Alcoholism and Alcohol Abuse, as well as from the William M. Keck Foundation. And then just as a final note here, I'm gonna come back onto Zoom and speak with you all tomorrow afternoon. But at this point, I'll be happy to take any questions that you might have.
Thank you so much for your insightful presentation. And I now open the floor for questions for Dr. Golding. And I do see a few questions on the chat already for you. The first question is from Dr. Dana Dolinoy. She asked, the mouse strain specific effects are interesting and concerning. Mm-hmm. Have you looked at molecular changes, including DNA methylation? I think you kind of addressed that in the talk, in the latter part of the talk.
So, so we've looked at DNA methylation in sperm, and we've looked at DNA methylation in the placentas, the C57 black 6 placentas, not the hybrids. And we've not seen any changes that I would consider to be, memorable. We do, however, see changes in a large number of imprinted genes. And so like H19 and CDKN1C, a couple of them from the KCNQ10T1 cluster. But as far as we can tell, these are not directly related to changes in DNA methylation.
Good. Thank you, Dr. Golding. I was struck by that strain-specific data that you showed, because we are involved, many of us on the Zoom are involved in a consortium called Target 2, which is looking at developmental environmental exposures. It's more of a maternal exposure, not a paternal. But when we did whole genome bisulfite sequencing, now this was on liver and blood, so not looking at the same tissues as you, the strain effects overwhelmed the environmental exposure effects. And so I was trying to get a little bit at whether there's something special about alcohol, or whether, versus a sort of mid-level environmental exposure, or whether we really do need to hone in on some of these strain-specific effects, which are usually ignored in rodents for epigenetics.
Yeah, so I got interested in this because there's right across the street from me is this guy by the name of David Threadgill, who has this collaborative cross, which is a very large outbred group of mouse models. And when he was working on a mouse mutant for the epidermal growth factor receptor, I think, he noticed that the strain had a dramatic influence on the penetrance of the either the embryonic lethal or the growth restriction phenotype. And so the changes in placentation between these little, these substrains of mice, particularly with that, with the labyrinth layer and the decidua, really seemed to explain, why it was that one strain could survive this genetic mutation, whereas another strain could not. So that's kind of what got me going on this line of thought in the first place.
Yeah, I can imagine being across the way from David Threadgill. Who wouldn't pique your interest in that?
Yeah.
That's great.
Yeah.
Thank you.
I see some raised hands for questions. So if you could unmute yourself and ask the question, that would be great. The first hand I see is from Stephen Krawitz.
Thank you very much. Wonderful talk. Really enjoyed every second of it, picking up some new ideas. One of the questions I had is, at least in humans, in the first 6 hours after fertilization, you have total replacement of the histones before sigamy. So how do you reconcile your effect, what you think is maybe a histone modification effect, when most, if not entirely, most, if not all, are replaced in such a short time?
Yeah, so that's a really interesting question, and I think it's one that we're struggling with. So there's some evidence that certain histone variants are able to persist through that exchange, but these data are all based on immunocytochemistry, and so I don't know how robust it is. But I think the idea there, the kind of larger picture that we're chasing here, is that these chromatin modifications might be in some way, shape, or form indexing other bookmarks that then read these regions here, and then in some way, shape, or form mark them for the memory then to persist. How that actually happens, I don't know. I mean, as probably you're well aware, that studying that very early developmental stage is incredibly challenging, especially with a large-scale genome-wide sequencing approach. However, Sarah Kimmon's group up in Toronto, I think, has done some work looking at folate deficiency, and she can track changes in sperm through into, I think it's the 8-cell embryo. And so she just had a paper published in Developmental Cell, I think within the last year, that described that. And so that's really been the first paper that I've seen that's bridged the gap between changes in sperm and changes in the early embryo. And so again, as a caveat, she could do this at the 8-cell stage, just because of the technical challenges, I think, of going earlier. But yeah, that's something that we're very aware of. And I think the short answer to that question is I don't know. And I think that these changes are relevant, but I don't have a good mechanistic explanation for how just yet.
And if I may, just one follow-up question to that.
Yeah.
So at least in humans, there are a series of about 100, 120 different messenger RNAs, which are delivered upon fertilization, which are full length and are nowhere to be found in the in the maternal repertoire.
Okay.
At least half a dozen are actually from the male X chromosome.
Okay.
So I just have you looked for anything like that yet?
No. So we've largely focused on the small non-coding RNAs and trying to make sense of that. There is a mouse paper that just recently sequenced the large non-coding RNAs as well as mRNAs that come in with sperm. It was in a completely different model, but so far we have not in the field have not really chased down that lead. So it's something that I think, that you've raised that point. I think it might be something we go back and start looking at.
So last question, and I truly mean last one, because your talk is fantastic. Have you ever then taken the males that have been on alcohol and then let them rest for 1 or 2 spermatogenic cycles?
Yes.
And then followed them through?
Okay, so we've done these experiments where we've looked at the non-coding RNA profile of the epididymis. And when they're on alcohol, we have a very strong alcohol profile. But when we pull them off alcohol and we wait for, I think it was 60 days, that profile reverts back to normal. And so it suggests that the males could recover from this. And so there's a caveat here that I'd like to throw out, in that these are adult males and we have never done an exposure during adolescence. And so my program officer is really keen to get me to modify the model to start looking at males as they start drinking through puberty. And this is a problem that they foresee might have a more dramatic impact on epigenetic programming. But these are all just, kind of thought experiments at this point.
Well, thank you very much.
Yeah, thank you. Those are great questions.
Thanks. I see another raised hand from Heidi, if you want to go ahead.
Yes. Hey, that was a fantastic talk. Thank you, Michael. So I thought the results with the sperm histone modifications were very interesting. However, the field has been kind of seen reports with various reports with different histone retention, already reported, different loci with histone retentions. Mm-hmm. And what people think is that, the way we isolate sperm from mice by, making the incision in the capsule and have them swim up, I assume that's how you isolated them.
Yeah.
Not all of that sperm has completed protamine to histone exchange.
Yes.
And so what those reports reflect might actually be the amount of immature sperm. So I was wondering if you think that the alcohol in males causes more immature sperm or causes the sperm to not complete histone retention. And that's why you're seeing such a really stark increase in H3K4 trimethyl in your Western blots even.
Yeah, so that, I don't know the answer to that question, but I think that the data that we have so far suggests that there's a problem with maturation. And so it's either in the non-coding RNA profile or in the elimination of histones and then replacements with protamines. I don't know if it's, one or the other or a combination of both, or if perhaps the non-coding RNAs are then targeting different regions for, histone removal. But this seems to be the line that I think is the most reasonable. So I don't believe that alcohol has a dramatic effect on 1-carbon metabolism, and that's the reason that we're seeing these changes in methylation, because, if it was true, it would kind of affect methylation across the board, and we don't see anything in DNA methylation. And so the other interesting point is that if it was inhibiting 1-carbon metabolism, we wouldn't have, the alcohol group completely swallow up the controls and then, have that dramatic increase. And so I think that it has to be either purposeful changes that are a symptom of alcohol, or it's a failure to completely mature. However, having said that, we don't see any differences in litter size, and the alcohol-exposed males tend to have, higher success rates in breeding. So it's not impacting fertility. And so I don't know if calling it sperm maturation is completely appropriate because it's not impacting the ability of the sperm to do its job.
Mm-hmm. Yeah, I don't think there's differences that they see when they try to, or when they artificially inseminate mice. I don't think there's a difference between that. A little bit immature sperm from the mature sperm. And then I've got one more question, just because I'm studying metabolism. Did you control for the calorie intake with the, feeding the males the alcohol spiked water?
No, we didn't.
Did you see any increase in weight, or is there any?
Yeah, so we never saw an increase in weight during adolescence. However, if we start taking them out into much later life, my students suggest that the alcohol exposed ones get a little bit fatter, but we haven't run statistics on that. So if there is a change, it's not dramatic, and it's certainly not during the period here where we're doing the breedings.
Okay, thank you.
Yeah.
We have one more question in the chat. It's from Raheem Lewis, asking whether you can explain where in your data is it clear that there is an association between the sperm of alcoholic male mice and fetal defects.
So I don't know. I think one of the problems with the field of epigenetics is that we try to attribute causality to an event. And so I think that, we do see changes in chromatin structure and non-coding RNAs in sperm, and then that correlates with changes in craniofacial development and then, placental and fetal growth. However, I think that these could all be symptoms of something else that has happened much earlier in development. And so I don't know that I could be able to trace the epigenetic change in sperm directly through and be a direct modifier of some of the other phenotypes. But I think that perhaps the more likely explanation is that the epigenetic changes in sperm are inducing some kind of stressor during development, and then these other phenotypes are emerging as a symptom of that stressor. Does that make sense?
Makes sense to me. I think Raheem's gone, so
Okay.
It makes sense to me. Thanks though, Mike.
Okay.
Mike, I have a question. This is Vasanta. I was coming from a functional end of it.
Yes.
I was thinking about all the changes that occurs in the placenta. Does that reflect in any kind of angiogenesis or placental transport to the fetus? To what extent those are contributing factors to some of these changes?
Right. So I'm a novice in placental biology. So I've done kind of the easy, large scale things, looking at weight and histology. We did see a large number, or a good number of placental transport proteins come out of our RNA-seq analysis, but I have not done the hardcore biochemical measures of placental transport and kind of these more classic studies that would get at the crux of what might be going on here to cause that loss and efficiency.
Does anyone have any lingering questions? Please feel free to ask.
I think it'll be really nice to actually pair them up, both the paternal alcoholism, maternal alcoholism, and see if there is a cumulative burden or, how the two things are different.
Yes, I agree. And so the hypothesis that we had going into this would be that that there might be a synergistic effect here if both mom and dad drank, but we didn't seem to see that, at least with the craniofacial defects. No, I mean, we saw them and they were perhaps more consistent, but in terms of their magnitude, there was not a synergistic effect. It was just, they were there kind of with the equal intensity, I guess, as the maternal alone or paternal alone.
Okay. That's great, Mike. Thank you.
Well, yeah, thank you so much for the invitation, and I guess I'll see you all tomorrow afternoon.